Welcome to International Conference on Basic and Translational Cancer Research, Novel Ideas and Approaches. Our speakers are Dr. Perya Swami, Dr. Raji Sundar Rajan, Dr. Ignacio Camarillo. Dr. Perya Swami is the Professor of Biology and Biomedical Engineering, Center Director of Center for Cellular Imaging at University of Virginia. He completed his PhD at Indian Institute of Technology, Madras, and his postdoctoral studies at University of Washington. Dr. Piria Swami is an internationally recognized expert in the advanced microscopy techniques, particularly in the area of molecular imaging in living cells, tissues, and animals. A key area of his research is focused on the design and development of optical methodology, including advanced light microscopy techniques, to investigate, monitor exogenous and endogenous protein-protein interactions, intravenous imaging, and monitoring the physical patterns of normal versus cancer cells. Dr. Perya Swami is the founder and center director of the internationally known Center for Cellular Imaging. He is one of the pioneers in the development of fluorescence lifetime imaging microscopy (FLIM) for measuring the oscillations in cytosolic and nuclear-free calcium in signal intact living cells. He developed a 2-3 color steady-state confocal multiphoton and film-based Foster resonance energy transfer (FRET) imaging system for protein localization in living specimens. He has published over 100 articles in referred journals and book chapters. He has given more than 140 invited lectures nationally and internationally. Dr. Perya Swami has edited 3 books series, and he is a series book editor on cellular and clinical imaging, and he has 6 books to his credit. He is the chairperson in organizing an annual international conference on multiphoton microscopy in the biomedical sciences through SPIE since 2001, and runs a hands-on training annual workshop on film and FRET microscopy at the University of Virginia. Dr. Perya Swami was elected as the fellow member of SPIE Optical Society in 2012. Dr. Raji Sundar Rajan is the professor at Purdue University. She completed her PhD in electrical engineering at Arizona State University. Her research interests include electrical and laser pulse mediated chemo drug delivery to cells and organs, electrical and electronics applications to medicine, healthcare, and home health gadgets, modeling and simulation of biological systems. It also includes the combining the big data analysis with new wireless patient data to help predict cancer. She had been honored with several distinguished awards and has international and national industrial ties and collaborations. Dr. Sundar Rajan is the member of several organizations including Electrical and Electronic Engineers, Bindley Bioscience Center, Purdue University, just to name a few. Dr. Camarillo is the Associate Professor of Biological Sciences at Purdue University. He completed his PhD at Wayne State University and his postdoctoral studies at University of California, Santa Cruz. Dr. Camarillo is an expert in animal physiology, endocrinology, mammary gland biology, breast cancer, and development of animal models of disease. His research focuses on dietary components and obesity on breast cancer progression, tumorigenesis, understanding the impact of diet and obesity on mammary tissue, and identifying key metabolic targets. He has used proteomic and genomic methods to identify mechanisms of leptin on tumor cell growth, epithelial polarity, and has developed a co-culture system to study molecular crosstalk between adipocytes and cancer cells. Dr. Camarillo has also developed a rat model of obesity and breast cancer and has shown that a Western diet can lead to differing effects on tumorigenesis and tumor phenotype based on adiposity levels. Dr. Camarillo is the first to use multiphoton multimodal imaging to characterize the influence of obesity on the mammary gland and tumor microenvironment. In his recent work, he has contributed to defining how leptin TAT3 signaling is involved in obesity-associated breast cancer progression. Further, he has used a rat model to show exercise during pregnancy can substantially decrease breast cancer development in female offspring. In previous work, he has employed a combination of mammary surgical procedures and mammary epithelial cells isolation techniques to characterize hormone receptor expression in developing mammary gland. Dr. Camarillo's expertise in the development of cell culture and animal models to study breast cancer progression places him in an excellent position to effectively complete the goals of the proposed studies. As a PI, he has trained 5 PhD students, 4 MS students, 3 visiting scholars, and over 50 undergraduates. He has won several honors and awards, including the Purdue University Top 10 Teachers in the College of Science Award.